Welcome to the next lecture of our course on environmental biotechnology, and in today's lecture we will be discussing on the microbial interactions with heavy metals and metalloids, particularly with reference to bioremediation. Now, in this particular class, the following concepts will be discussed. We will discuss about microbial interaction with metals that facilitate bioremediation. We will learn about some of the important aspects of biosorption and bioaccumulation, biotransformation that facilitates metal precipitation and metal solubilization, both by dissimilatory as well as enzymatic reduction processes will be discussed. This will be followed by biomineralization and some points about bioremediation strategies. Now, microbe metal interaction is considered to be the key to the metal bioremediation process. Now, with respect to bioremediation of heavy metals, including the metalloids and also the radionuclides, microorganisms cannot destroy the metals, they are not alchemists. So, this is a very fundamental point that we all must understand, and this understanding helps us to actually realize that then how microorganisms could be useful in developing bioremediation strategies, because metals will remain metals even after the microbes they interact with themselves. So, there must be some kind of very intelligent way of looking into this interaction where or interactions, because microbes interact with a number of processes. These processes through which microbes interact with different heavy metals, metalloids and radionuclides, it is truly very diverse, and this diversity of interactions in many cases what we see that can only alter the chemical properties, sometimes making them more available to the rest of the ecosystem components, sometimes allow them to precipitate, sometimes allow them to be more solubilized, or sometimes allow these metals to be sequestered or absorbed or accumulated within some microbial cells like bacteria or fungal cells. Now, some of these processes of interactions can be used to treat the metal contamination. Now, when we say that treat metal contamination, so this could be practically all type of metal contaminations, whether it is only the metals present there or metals present along with hydrocarbons and other pollutants. So, over the periods of time, like we see that for last 30-40 years, our understanding on this microbe metal interaction has improved to such extent that now we are in a position to better comprehend the entire process of this microbe metal interaction and then develop strategies in which these microbial interactions with metals can be utilized for the bioremediation process development. Now, microbial interaction that help in treatment of metal contamination could be of three basic types. Now, microbial processes that eventually alter the chemical properties. Now, we say chemical properties, that means it also affect the environmental behavior or environmental fate, environmental toxicity, bioavailability of the heavy metals. So, these are a wide ranging processes. So, these are multiple processes which are involved in altering the fate and chemical properties of the heavy metals. So, the first and foremost is the highly specific biochemical pathways which are present in microorganisms that have evolved to protect the microbial cell from toxic heavy metals. Since microorganisms are living with these metals from the origin of the life or origin of their systems, so they know, they have learned how to deal with these metals. So, many of these metals like chromium, uranium, cadmium, mercury, most of these heavy metals, arsenic, they are toxic either at low concentration or at moderate concentration or even at high concentrations. So, no matter what is the type of metal? Most of the metals are toxic, or even all metals are toxic beyond certain threshold limits that we all know. Even the trace metals, which are essential, considered to be essential for microbial metabolisms, they are also toxic beyond certain threshold level. And also the metals which are not absolutely required by living cell, like for example mercury, arsenic and cadmium for example, they are toxic to even at a lower concentration. Now, for all the metals, all different types of metals, microbes have developed different detoxification mechanisms. These detoxification mechanisms are one of the targets during the bioremediation process development. For example, the chromium detoxification, arsenic detoxification, mercury detoxification, or even the cadmium or cobalt or copper detoxification system. These are naturally present in microbial cells, microbial system. So, what we are doing, we are learning these detoxification mechanisms and trying to improve it, make it more specific, and then trying to see that how these detoxification based metal transformation or metal detoxification processes can be developed, so that they will be suitable for metal bioremediation system. The second one is the fact that microbes can produce new mineral phases via non-specific mechanisms that result in the entrapment of the toxic metals within the soil or the sediment. So, these are mostly non-specific interaction, non-specific in the sense that they do not discriminate between the exact name or the type of the metals. These interactions are mostly targeted towards a broad categories of metals like divalent cations for example. So, many of the divalent cations or positively charged redox species are subject to a specific type of interactions, and these interactions with the microbes eventually led to the entrapment of the toxic metal in some component of the environment. And if it is within the soil or sediment or the mineral phases, then it is good for the bioremediation purpose. The third one is the production of biogenic ligands, which are chemical moieties or chemical groups that can complex the toxic metals, resulting in their mobilization or sometime immobilization from the contaminated soil. So, this is a kind of these biogenic ligands like phosphate, carbonate, hydroxyl or sulfide, these ligands which are produced by microbial metabolism, they interact with the metals which are present in the environment and then alter the nature of the metal. Sometimes they make the metal more mobile, more soluble, sometimes they make the metal insoluble. If they are insoluble, products are insoluble, then the insoluble metals are precipitated, and that becomes direct benefit out of the interaction, because precipitation leads to their local immobilization of the metal. Whereas, the mobilization of the metal, sometimes the organic ligands particularly, when they are attached to the metals become more soluble. So, these soluble metals are found to be very useful, because these soluble metals can be pumped out of the sediment or leached out of the sediment, and then they can be trapped in a bioreactor on the surface and then can be reused in certain other industrial process. So, both these mobilization and immobilization processes, which are facilitated by different biogenic ligands, are found to be useful. So, in a nutshell, what we see that there are three distinct, there may be many processes that I will discuss. There may be many processes through which the microbes interact with the heavy metals and metalloids and radionuclides, and those processes can be suitable for metal bioremediation, but all those different processes can be categorized into three broad categories. One is the detoxification system based, another is the entrapment based, and the third one is the mobilization or immobilization based through biogenic ligands. Now, if we see this particular schematics, so here the diverse array of microbial interactions with metals with potential for bioremediation are discussed. So, this is the microbial cell, and I have just enlarged the cell in these two other live boundaries in order to accommodate different type of interaction, different types of interactions. So, first is, as we discussed in our earlier lecture, that the divalent metal ions or maybe other metal ions also like arsenate or chromate can also be transported inside the cell. So, the microbial cells, they are able to accumulate the metals inside the cell. So, toxic metals can be accumulated inside the cell. So, this is one way of interaction. Now, since the metal is toxic, or many of these metals including the chromate or hexavalent chromium, the chromate, or arsenate, where arsenic 5 plus, all these are toxic. Even the cadmium, cobalt, zinc could also be, cadmium is anyway toxic, but the cobalt, zinc, nickel, etc., which are non-toxic at low concentration, but could be toxic or are indeed toxic at higher concentration. So, microbes will be utilizing different detoxification strategies obviously, but before deploying the strategies, detoxification strategies, the microbes will accumulate metal. So, it is not that all microbes will accumulate all metals, of course, but what we see that many microbes are capable of accumulating the metals, particularly up to certain concentration levels, the divalent cations and even arsenate and chromate, they are also accumulated inside the cell. This is called a bioaccumulation based process, which is interestingly driven by the metabolism. Bioaccumulation is of course driven by metabolism, because when we look at the bioaccumulation process, we will be able to see that there are involvement of specific transporters. So, these are the specific transporters. So, this is for example a specific transporter which is called metal transporter, for example, and metal transporter often they require supply of energy like ATP, because these processes are energy driven process. So, these processes are most of the time we found that these are the characteristics of the metabolically active cells. Why metabolically active cells? Because metabolically active cells will only have sufficient energy, for example, in the form of ATP. Now, the second one is the biosorption, which is basically the adsorption and absorption of divalent cations and on the cell surface mostly, and this is a metabolism independent process. So, no energy is involved in this. So, this is almost executed for all the divalent cations and almost all microbial cells, gram negative, gram positive, fungal cells, algal cells, all cells they show these properties because of the anionic nature of the cell membrane and cell envelope, while the divalent cations they found it's a nice place to bind on those the anionic or negatively charged groups. The next process is an enzymatic or otherwise transformation process, which is called a bioreduction process, where the soluble metals could be different types of soluble metals are there, for example, uranium. The soluble metal which is in oxidized form can be reduced further, and following reduction the insoluble species will be produced. So, following reduction the insoluble species will be produced, and then the insoluble form will precipitate. So, this insoluble form will be precipitated, and this is catalyzed by specific enzymes. So, these enzymes may be very specific to the metal concerned, or they may be coupled with the dissimilatory process. Dissimilatory, I will discuss this thing in little more detail. So, these processes are catalyzed in one case by specific enzymes, like we will discuss that for example, mercury or for arsenic. We can see, because these arsenic and mercury are absolutely not required by the cells. So, but they are toxic. So, arsenic and mercury type of heavy metals or metalloids can be reduced by bacteria using specific reductases. So, these are considered as enzymatic reduction, whereas for example, the chromium, uranium, technetium and several other radionuclides and metals, they are often used as electron acceptor during the anaerobic growth, anaerobic metabolism of the cells. And during this anaerobic reduction of the chromium or uranium or technetium or similar other heavy metals, the microbes are not interested in the reduced product. That is why it is called dissimilatory. Dissimilatory in the sense that the reduced products are not utilized by the microbes. Otherwise, if the reduced products is used by the microbes, then it will be considered as assimilatory process. So, the bioreduction could be enzymatic and dissimilatory, and the reduced product is insoluble. So, basically this will precipitate, that this will not remain in the solution phase at all. So, the next process is a very interesting process which is called biomineralization. During biomineralization, the microbes, they produce different type of ligands. One of the very interesting ligand is the phosphate group, which the microbes often produce because they break down or they utilize the organic phosphates and the phosphate groups are released. And these phosphate groups are very efficient in binding the divalent metal ions, and they can lead to the precipitation of the metal phosphates, and all the metal phosphates, they precipitate in the surrounding environment. So, around the cells, outside the cells, these metal phosphates are precipitated. So, these biomineralization mostly facilitate the precipitation of the insoluble metal deposits, which are either with phosphate or with sulfides, and they precipitate in the outside the cell. Similarly, the bioreduction is also a process which reduces the metal in the outside the cell. So, the insoluble metals will be deposited or precipitated outside the cell. The cell will not be accumulating those reduced or mineralized product in most of the cases what we see. Similarly, the sulfides which are produced during the sulfate reduction. So, this is another process which is mainly carried out by anaerobic bacteria, anaerobic sulfate reducing bacteria. They use sulfate as the terminal electron acceptor, and they reduce the sulfate as their terminal electron acceptor, and sulfate is converted to sulfide. And this H2S, which is basically gaseous H2S, it has the ability to interact with the divalent cations which are present in the vicinity, and it is found to be one of the best biomineralization method, because the moment the sulfate reducing bacteria, they are capable of producing the H2S, the H2S readily reacts with the divalent cations and produces the metal sulfides. Now, metal sulfides are again insoluble and they form the minerals. So, from their small nucleation centers, gradually the minerals, they grow up, and a huge deposits of metal precipitates, metal sulfide precipitates or metal phosphate precipitates, they form. Now, all these mechanisms, bioaccumulation, biosorption, bioreduction and biomineralizations, they have been found to be very useful for bioremediation or for the removal of the metals, because in all these cases we find that they either remove, like biosorption or bioaccumulation, they remove the metals from the aqueous phase, or the bioreduction that allows basically the precipitation of the insoluble metal, or the biomineralization that also facilitates the precipitation of the mineral form of the metals. However, through the production or use of some ligands which are produced by the bacteria or the fungal cells. Now, here are another interesting way of dealing with the metals which are executed by many bacteria and fungi, that is the production of organic acids. Many microbes, they produce organic acids which can react with the insoluble metals, and when the insoluble metals are reacted with the organic acids, so the metal organic complexes are formed, and those complexes are found to be mostly soluble complex, and these soluble complexes are more mobile than the insoluble counterpart. Therefore, the soluble metal complexes, they leach out or they mobilize from the soil or the sediment, and that process is called bioleaching. During the biohydrometallurgy or recovery of different industrially important metals, this process of bioleaching has been found to be very important, and we will discuss about this bioleaching in a separate lecture. Now, the another mode of the redox transformation is called biooxidation. In case of biooxidation, what happens? The metals which are reduced and insoluble, they are oxidized to produce more soluble form of metal. And these more soluble forms of metals, even they also help in the bioleaching or extraction of the metals from the contaminated soil or contaminated sediments. So, often if we want to flush out or even we want to recover or remove the metals from the sediment or the soil or other environments, we can use this bio-oxidation or the organic acid mediated bioleaching process, where the metals will be solubilized and the soluble metals will be mobilized or will be removed from the solution or the sediment, and then they can be either precipitated by some other means and can be recovered and reused. Now, there is another very interesting method of bio-oxidation that has gained considerable attention in recent time, that is called bio-oxidation through dissimilatory mode of interactions. So, metals like manganese, for example, which when they are in reduced form, they are more soluble. So, manganese, for example, manganese 2 is soluble, more soluble when they are reduced form. But if we are able to oxidize them, then it is converted to Mn4, that is MnO2, and this MnO2 is readily precipitate, they form actually minerals, and one of the concepts of biomineralization could be by bio-oxidation of iron or manganese kind of things, which can be oxidized through microbial redox transformation. So, these are again redox mediated transformation that microbes can use these, and then they convert these manganese or iron, for example, into insoluble forms, and these insoluble forms they precipitate and they can be useful for further bioremediation process. So, that I will discuss. Now, once metals are in solution, one of the simplest way to remove them is through the bioabsorption. So, we will now go back to the processes which are just discussed, like bioabsorption, bioaccumulation, followed by the precipitation based methods, and then again the biomineralization based processes. So, the bioabsorption process has gained considerable research interest at some point of time, because it was found to be the simplest way to remove the metals and radionuclides, because it is metabolism independent often, and the microbial cells are having high surface area to volume ratio. They can interact with these divalent cations, for example, very rapidly, and they can sequester or they can absorb these divalent cations very effectively. So, these are metabolism independent absorption process and mediated by the cell surface negative charge groups like the carboxyl, amine, hydroxyl, phosphate, sulfahydryl groups, and can absorb appreciable quantities of positively charged cationic metals. So, huge amount of metals can be precipitated or can be absorbed, rather. We do not use the term precipitated in this case, we say it is absorbed on the surface, because it is not an either enzymatically mediated precipitation nor ligand based precipitation. So, here as you can see that the cell envelope of organisms, different type of organisms, they have been found to be very useful in having this negatively charged moieties or ligands in the cell membrane or peptidoglycan or other capsular structure of the cells. They are able to bind the divalent uranium, for example, in this case. So, divalent cations can be adsorbed through electrostatic absorption through these anion groups, and then subsequently microprecipitation and ion exchange process can also take place, and eventually more amount of the divalent cations can be deposited in the absorbed on the cell surface. And since it is a metabolism independent process, it has gained considerable interest, because the toxicity and all those points are not valid, because these cells may be dead also. So, basically dead cells can be used for this purpose. On the contrary to this, bioaccumulation, which is very well studied in case of Pseudomonas and other species, have been found to be facilitated by internal transport, and through different type of metal they have the property to penetrate the cell, like uranium is a very toxic, and they have the ability to penetrate the membrane and enter inside the cell, and there the cells are able to precipitate or sequester this internal uranium UO2 plus through polyphosphate granules, because the cells they need to have some mechanism to combat the toxic effect of uranium. So, the bioaccumulation, as I mentioned, as long as the concentration is within the tolerable range of the organisms will be happy, but as soon as the concentration of the internalized or the bioaccumulated metals go beyond the threshold, the tolerable limit, then the bacteria or the fungi, they will express, then they will execute different detoxification strategies. So, we have noticed that for not only for uranium, for a large number of other heavy metals, the microbes produce a significant number of detoxification strategies, including efflux, as we discussed in our earlier lecture, or expression of metallothioneins in eukaryotic systems, or even in cyanobacteria we see small cysteine sulfahydryl group containing protein molecules for sequestering these toxic divalent cations, cationic heavy metals or radionuclides inside the cell. Now, the advantages of these bioabsorptions are the bioabsorption methods seem to be more effective than their physicochemical counterparts, because for many waste including uranium also, physicochemical methods including other divalent cations, cadmium, zinc, etc. for industrial waste treatment, physicochemical methods including the resins, etc., ion exchange resin are in use. So, bioabsorption process actually was developed the kind of a and it's kind of a comparative technology, comparable technology, which can offer a solution to the removal of the metallic pollutants, because it can remove the dissolved metals at very low concentration. That was one of the best advantages, that below 2 to 10 milligram per liter the bioabsorption mechanisms can work very effectively. And I must say that ion exchange resins are not suitable for such low concentration. Otherwise, the cost will be very high if we want to use some physicochemical methods for very low concentration of waste. Potential use of low cost waste biomass. So, many other industries including the biotechnology industries, often they produce the for example, the brewery yeast and other systems produce large amount of biomass. So, those biomass can be utilized for bioabsorption processes. Even the dead biomass can also be used, and dead biomass is found to be a better biosorbent, because the metabolism is not involved and toxicity problems are not associated with dead biomass. Bioabsorption shows basically rapid kinetics and high adsorption capacities. It is also possible to remove the bound metals from the cell surface to regenerate the biomass. So, often we have seen that the biosorbents can be used several cycles, like we can rejuvenate the biosorbents after we remove the metals, bound metals from the cell surface. Higher specificity of biosorbents may avoid an important problem encountered with ion exchange resins, and with particularly ion exchange resins having the problem of the overloading of the binding sites by alkaline earth metals present in the polluted environment. But in some cases we have found that the biosorbents, algal and fungal biosorbents, bacterial biosorbents are found to be more specific to certain heavy metals and radionuclides. So, that is again another advantage against the ion exchange resins. Further, the biological systems, since the biosorbents are basically obtained from biological systems, they are bacterial or fungal cells or algal cells. So, we can actually engineer the cells or genetically modify the cells to improve their specificity and affinity. Now, there are some significant problems associated with this bioabsorption, like the desorption from the cell surface can be as rapid as absorption. Sorbed material could be released to solution when cells die and decompose. Sometimes they disintegrate also, releasing the metals into the solution again. Other cations compete for binding sites. So, all biosorbents are not very specific. So, there may be some competing ions are there, and those competing cations can bind and enforce some kind of competition with the target toxic metals. Cell surfaces can also quickly become saturated, preventing further bioabsorption. And essentially, bioabsorption is considered to be not an adequate long term solution for in situ bioremediation. So, it may be suitable for some of the pump and treat scenario, where we can have a column based process, and through that a number of large number of bioabsorption based processes have been actually developed. Now, in the next section, we are going to discuss about the genetic engineering of the bacterial biosorbents, particularly using the concept of the metallothioneins, which are found to be low molecular weight cysteine rich proteins engaged in binding toxic heavy metals in mostly in eukaryotic system, but also present in cyanobacteria. So, these metallothionein genes are expressed in bacteria. For example, in experimental way, we express them in E. coli, and when we express the eukaryotic metallothionein in E. coli through genetic engineering, we found that they can efficiently bind cadmium, accumulate cadmium. It is a part of bioaccumulation. E. coli accumulates the toxic cadmium, and E. coli cells can sustain the toxicity because the cadmium ions are bound with the metallothioneins, which are expressed inside the recombinant E. coli cells. In order to circumvent the problem associated with the cytoplasmic expression, that is the metal uptake limitation. So, if we want to have cytoplasmic, like in this case cytoplasmic expression of the metallothionein, the metals need to be transported inside the cell. So, you have other issues also. So, the scientists, they thought that instead of expressing the metallothionein in the cytoplasm, if we just express the metallothionein on the periplasmic space on the outer membrane, that might actually relieve the cells in terms of any toxicity issues. So, that was also done successfully, where the metallothioneins were expressed in such a way that they will be located on the periplasmic space or the outer membrane and or the membrane envelope, and the divalent cations like cadmium can be effectively bound to this. Now, this is not end of the road with respect to metallothionein based engineering. More specific and cytosolic metal removal systems were further developed through engineering the transport process. So, we also engineered specific transporter like nickel transporter, and the nickel is transported inside the cell, divalent nickel ions, and then the nickel ions were precipitated or accumulated through the use of the metallothioneins. Now, with respect to metal precipitation, which has been found to be one of the major means of bioremediation of toxic metals and radionuclides, we will discuss this part in a little more detail. So, fundamentally, the microbial cells are capable of precipitating metals like uranium, for example, where the soluble metal species like UO2 plus is reduced to insoluble metal species like uranium 4. So, uranium 6 to uranium 4, and the uranium 4, this particular form of uranium, it precipitates. Naturally, it precipitates in an aqueous environment with near neutral pH. Now, this particular process of metal precipitation through redox transformation, basically we are transforming the oxidation state of the metals or the radionuclides, can be achieved by two means. So, bacteria can be used to efficiently immobilize, that is, the precipitate the metals or radionuclides, because of their capacity to reduce these metals in a lower redox state, like in case of uranium, we see uranium 6 to uranium 4, producing less bioactive metals. So, uranium 4 is not at all bioactive because it precipitates. This precipitation can also be achieved through inorganic ligands produced, that we have learned during our biomineralization discussion, that by producing different inorganic ligands like sulfides, hydrogen sulfide or phosphate or even carbonate, the microbes can also lead to the precipitation of the metals, like even uranium can be precipitated by uranyl carbonate or uranium phosphate, and many other heavy metals can be precipitated through sulfides precipitation. Now, these are the two processes that you can see here, that for the bioreduction, that is the first one, that they reduce the elements to a lower redox state like uranium 6 to uranium 4. So, you have the uranium 6 that can be reduced to uranium 4, and for this reduction process you need a source of carbon and source of electrons. So, that can be organic carbon. So, ideally these organisms will be mostly heterotrophic, and they can engage either a cytochrome based system or a flavin based system. So, there are multiple systems discovered through for the transfer of the electrons from the electron donor to the uranium UO2 2 plus, so that the UO2 2 plus can be reduced to different form of uranium which can precipitate, which are insoluble. So, effectively this type of reduction is found to be possible for not only for uranium, but also for chromium, for technetium, for selenium and many other similar redox active metals where the reduced forms are insoluble. So, this is found to be one of the best way even for in situ bioremediation, because it has been found, and I will discuss that briefly, that by providing suitable carbon source, and if you have this appropriate microorganisms who can do these things like use their cytochromes or flavin based systems to transfer the electrons and reduce the uranium species or chromium species like hexavalent chromium to trivalent chromium, or uranium 6 to uranium 4, and then uranium 4 can precipitate or will precipitate in the exterior environment, and that will remain in a precipitate form unless and until that is going to be reoxidized. So, that part I will also talk briefly. Now, the other part that I mentioned is the precipitation of the metals through inorganic ligands, which is a part of biomineralization as well. So, here you can see that uranium, for example, can be biomineralized because by producing phosphates. Now, these phosphate moieties can be produced by providing glycerol phosphate. This is the organic phosphate donor. This organic phosphate molecule can be metabolized, can be broken down, so that the phosphate groups are released by bacterial phosphatase. So, specific bacterial strains have been isolated and characterized who produce this phosphatase enzyme, and they can also allow this precipitation of huge amount of uranyl phosphate, and not only uranyl phosphate. So, large amount of metal phosphates are produced, and when they start depositing nearby the cells, they form a nucleation center, and as the nucleation of this uranium phosphate or other metal phosphates starts, this nucleation center basically allows the further growth of the minerals, and then a chemical deposition of these uranyl and other metal phosphates are formed. Now, this process of metal reduction leading to the formation of the insoluble or immobile reduced forms, which have been found to be very popular and very effective for heavy metal and radionuclide bioremediation, can be achieved by two mechanisms. One is called direct enzymatic reduction, where the microorganisms involved allow the flow of electrons from the organic compounds and reduce the uranium, chromium, technetium, etc., which are present in the oxidized form, and in all these oxidized forms are more soluble, more toxic, and maybe in the groundwater, in a soil or sediment if they are there. Then if you are able to provide the suitable electron donor and carbon source and your appropriate microorganisms are there, then they will reduce it through these cytochromes or flavin based systems, and it is called a direct mechanism, because direct enzymatic reductions, they allow conversion or the reduction of these highly soluble species into insoluble species, and these insoluble species they readily precipitate into the surrounding environment. The other process is called the indirect enzymatic reduction, like the Sivanella type of organism who can actually reduce the iron Fe3 to Fe2 by iron reductases, and Fe2 can further reduce the uranium, technetium or chromium. So, it is indirect reduction by using Fe2 plus as the electron donor. So, Fe2 donates the electron and uranium, chromium or technetium is reduced. So, thereby it is called indirect enzymatic reduction. Now, indirect enzymatic reduction also occurs in case of sulfate reducing bacteria like Disulfo vibrio and many other species, where the sulfides which are produced, these bacteria can also use the like periplasmic hydrogenases and can actually facilitate the precipitation of soluble uranium into insoluble deposits. Now, with respect to biomineralization by ligands, this process is basically a process in which metals are precipitated with microbially generated ligands like phosphate or sulfides. And these are one of the best examples is the Citrobacter species, and this particular Citrobacter species has been studied very elaborately with respect to its ability to produce this phosphatase enzyme, and this phosphatase enzyme can precipitate different type of, allow the precipitation of different heavy metals including radionuclides also in a very large quantities in and around the cells, provided a organic phosphate donor is provided. So, the phosphatase enzyme basically catalyzes the release of phosphate moiety, and the phosphate moiety is able to react with the metal. Again, this is to some extent indirect mechanism, because the microbe is basically dealing with the phosphate, and the phosphate group with ligands which is produced is reacting with the metals. So, similar things are also there with the sulfide producing sulfate reducing bacteria. The sulfides which are produced, they react with the metals, divalent metals, and allow the precipitation of the metal sulfides. For example, in the right side there is a picture of the uranium phosphate biomineralization. You can see around the cells large amount of uranium phosphate depositions are reported in a recent paper by Newsom et al. And in a further study we have found that this group of researchers like Meleke in 2019, they have found that europium, another very interesting rare earth element, is biomineralized in a similar way in and around the cell. So, you can see this is the bacterial cell, a large amount of europium is deposited, and through a very sensitive method of energy dispersive X-ray analysis, the scientists have confirmed that this is the europium deposits. Now, enzymatic transformation of metals and metalloids as part of the detoxification strategies. So, now these are enzymatic processes which are basically not connected to electron transport or dissimilatory type of reductions, like we have learned just before this that uranium, chromium, technetium etc. are reduced as electron acceptor. But other than that, some other toxic metals or metalloids can also be transformed enzymatically. These enzymatic transformation also lead to the conversion of the metals or metalloids into less toxic form or an altered form, which can be then utilized in the bioremediation process. And mercury and arsenic are the elements for which these reactions are best studied and implemented also for bioremediation processes. Now, we should remember that mercury is not essential anyway for the cell, and that is true for the arsenic also. So, it is absolutely clear that mercury enzymatic transformation of mercury or enzymatic transformation of arsenic by microbes are purely detoxification strategies. So, for example, resistance to mercury is very well studied, because this is catalyzed by the cytosolic enzyme mercury reductase. So, you can see here there is a MAR-A, the name of the protein is MAR-A, which is mercury reductase, and once the mercury is transported inside. So, for mercury, most of the mercury reducing bacteria, they possess the complete set of genes, that is called operon, and they transport through specific transporter. So, mercury is very toxic. So, divalent Hg2 plus is very toxic. So, you need to have a very specific transporter, so that the Hg2 plus enters inside the cell, and then immediately it is reduced to Hg0, and Hg0 is a volatile form and it leaves the system. Similarly, using arsenic also, so I will talk about that, but before that, this based on this mercury reduction system, one of the pioneering work was done with the radio resistant Deinococcus radiodurans for in situ remediation of a highly radioactive environment where lots of mercury is there. So, there are certain such contaminated sites for which this engineered Deinococcus radiodurans was developed, and with mercury reductase ability. So, the Deinococcus radiodurans is a natural bacterium who can withstand high amount of ionizing radiation, and still when it is engineered in terms of mercury reduction, it is capable of reducing mercury as well. Now, with respect to arsenic, we find that there are a number of strategies. Now, arsenic is a very interesting metalloid for which we have two forms. One is arsenic 3, which is more toxic, and we have arsenic 5, which is less toxic. Now, arsenic 5 enters inside the cells through phosphate transporter, because arsenate resembles phosphate and enters into the cell by mimicking the phosphate, and once it enters into the system or the cell, In the cytoplasm, this enzyme which is encoded by ARSC, that is a cytosolic arsenate reductase, it reduces arsenate to arsenite. So, AS3 is produced. Now, AS3 can also be transported inside the cells through specific proteins or proteins which are called aquaporins, kind of porins, which have been reported to be involved in arsenic transportation as well. This could be a part of the arsenic homeostasis, what we called, that cells they try to make living with arsenic. So, they do not say no to the arsenic, they allow the arsenic to come in, and all the arsenic 3 which are accumulated because of either direct entry through this aquaporin kind of pumps or by reduction of the arsenate to arsenite are subsequently effluxed out by different efflux system like ARSB system or ACR3 system or other systems which actually utilizes different type of energy sources, and they throw out or efflux out the arsenic as arsenic 3. Now, apart from these, microbes are also capable of transforming arsenic, like arsenic 5 can be reduced to arsenic 3 plus by what you called periplasmic reductase, and which are often connected to the dissimilatory process also. So, recent time we have seen that many bacterial strain they are capable of doing this reduction using the cytochromes and similar flavin based systems which we could notice for uranium as well. So, there are these bacteria under anaerobic condition. Under anaerobic condition, they use arsenate and reduce arsenate to arsenite, and since arsenite is more soluble, it goes into the aqueous phase, but arsenite is more toxic also. So, the microbes they must utilize some strategy to combat the arsenic 3 toxicity. For that, they use another periplasmic space or cell envelope based enzyme, that is called AIO or arsenite oxidase. So, this arsenite oxidase it converts. So, this is basically we called it AS3 plus oxidase. So, these arsenite oxidase is very specific enzyme, it is not a kind of a general cytochrome or flavin based enzyme. This is very specific enzyme arsenite oxidase, and those bacteria who possess these arsenite oxidase in their outer membrane, they are capable of oxidizing AS3 plus to AS5 plus, and this could be considered as a detoxification strategy again, because arsenic 3 is toxic, very toxic, and if you do not oxidize it to arsenic 5, then it will enter through this aquaporin and come inside the cell, and then within the cell it will execute toxicity, because arsenite 3 will interfere with the cellular sensitive macromolecules. Otherwise, you have to use some efflux pump to throw it out. So, some bacteria they adopt this mechanism that allow it to come in and then efflux it. Otherwise, some bacteria they prefer to oxidize it to AS3 plus. So, these are found to be very interesting mechanisms which actually allow the cellular homeostasis in terms of arsenic. Now, towards the bioremediation strategies, all these microbe metal interactions, almost all the types that we discussed today, they are tested up to field level also for in situ or ex situ treatments. So, one of the systems was this Mender system. That system is basically one of the best established at some point of time for in situ detoxification of metal polluted effluents, particularly with bacterial, algal and higher plants consortium, and the system were able to remove the divalent cations with 99% efficiency. Similarly, different consortium based system where multiple microorganisms are there and they are involved in metal removal through multiple processes including precipitation, biosorption and particulate entrapment, also allowed the development of different metal bioremediation, particularly with the use of or the role of sulfate reducing bacteria in the sediment system. Genetically engineered metallocyanins, they have been found to be very important in soil remediation, and particularly in one of the cases we found that Ralstonia eutropha CH-34, one of the best known metal resistant bacterium, is engineered with respect to this metallocyanin gene, eukaryotic metallocyanin gene, and the metal tolerant, this is naturally metal tolerant, this metal endurance CH-34, which is also known as Ralstonia eutropha, is modified with the mouse metallocyanin to in a surface to promote the metal bioabsorption. So, the bacterium was engineered so that on the surface of the bacterial cell, the divalent cations like cadmium, for example, actually the experiment was done with cadmium. So, in a cadmium polluted soil, if they try to grow the tobacco plant and the plant is killed due to the toxic cadmium toxicity, and when normal Ralstonia eutropha CH-34, it is not engineered, was added, bioaugmented rather, poor plant growth was observed. However, when the soil was augmented with Ralstonia eutropha CH-34, which is engineered with respect to metallocyanin genes, it has been found that the normal plant growth is there. So, that proves that the metal immobilization by bacteria could be potentially having a long term effect in reducing the metal bioavailability, because the metals seem to undergo a degree of precipitation around the bacterial cells, particularly this type of cells, and metal immobilization in soil using this type of system could have a long term effect, but unlike addition of the chemical amendment like zeolite etc., microorganisms could be employed to immobilize metals in moderately polluted fields, thus allowing their use in agriculture. Inoculation of metal resistant bacteria like bioaugmenting this type of engineered or otherwise naturally present bacteria who can actually sequester or bioabsorb huge amount of metal externally, found to be very efficacious in terms of protecting the soil which are moderately contaminated, and they have been found to protect the indigenous bacterial community from the heavy metal toxicity also. Now, since heavy metals usually coexist in waste with other kinds of contaminants, particularly organic pollutants, so lots of development have been done on simultaneous detoxification of the multiple pollutants, particularly the organic pollutants as well as the heavy metal pollutants. For example, Microbial consortium was developed which can oxidize organic pollutants like phenol, and utilizing that electrons releasing from the phenol oxidation and using that carbon, they can facilitate the chromium reduction. Development of strain that can detoxify both mercury and toluene in a radioactive waste, particularly with the Deinococcus radiodurans, and using the hyperthermophilic bacterium pyrobaculum, which can actually reduce uranium, chromium, cobalt, technetium using hydrogen as the electron donor was found to be very effective. And we can see that this in situ long term reductive bioimmobilization of chromium in groundwater using hydrogen release compounds. So, they use some hydrogen release compounds and they were able to implement on a field level this groundwater bioremediation of chromium. Similarly, the uranium reduction ability of the microbes, because I have discussed in detail about uranium reduction, that is also that is also used significantly in bioremediation of uranium contaminated groundwater in different subsurface environments, particularly the US DOE sites. And as you can see, there are two types of systems. In one system, the indigenous microorganisms who are there, they are stimulated to reduce uranium, because uranium can be used as electron acceptor, and by providing a suitable carbon and electron source like acetate, the acetates are injected. So, you have an injection well. So, the injection well basically in the underground environment, this carbon and electron donors like acetate is injected, and utilizing that the microbes are able to precipitate uranium, and these are insoluble uranium. Now uranium, when it is reduced, it will be precipitated, but the reduced uranium may be reoxidized to uranium 6 again following cessation of the organic carbon injection, accompanying subsequent delivery of oxidant like oxygen nitrate and iron Fe3+ in the etc. So, in order to reduce the chances of reoxidation of uranium from the reduced from the oxidized from, the scientists they found another innovative way that if they are provided with Fe3, then Fe3 might be reduced to Fe2 and the ability to produce these iron sulfides or iron minerals will effectively actually create a kind of a low permeability conditions or coatings, and these low permeability conditions will actually stabilize this uranium, allowing it to remain in the solid phase. So, it will in a sense will immobilize the uranium 6 other uranium 4 I am sorry. So, otherwise it would have moved out. Now, the last point in today's lecture is the biogenic metals, because in my summary slide I showed that the microbes can actually oxidize metals like manganese and produce manganese oxide minerals. Now, these manganese oxide minerals, which are called biogenic minerals, can be utilized in treating wastewater and water, particularly when they are having emerging pollutants, including some of the estradiol or other pollutants, and they can also allow removal of arsenic like arsenic 3, because oxidation of arsenic 3 can be possible by these biogenic manganese oxides, or oxidation of organic pollutants even in micro quantity can be possible by biogenic manganese oxides. So, in a recent time we see increased research interest is shown towards developing bioremediation technologies using these kind of biogenic minerals with manganese or even iron. So, for this lecture these following references can be used, and in conclusion, in today's lecture the details of the mechanisms and importance of metal microbe interactions in metal bioremediation is discussed. Scope of improving microbial metal binding, accumulation, sorbing capacities through the expression of metallothioneins and specific metal transporters are highlighted. Mechanism of bioremediation through precipitation and other processes are explained. Thank you.